Good evening, everyone. Welcome to Byju's Exam Prep Bias. First and foremost, if my audio and video both are fine, please let me know. Good. All good? Yeah. Right. So, in the last class, we had discussed some of the important portions about biotechnology. We discussed some of the application areas of biotechnology, plus some of the specific topics like what is cloning and what is stem cell therapy, etc. All these that were recorded in the last class. So I hope that every one of you have gone through that class. I could not take yesterday's class live because there are some problems because of which I could not come and take the class on time. That's why I pre-recorded class in the afternoon itself instead of the evening. So I hope that you've gone through the class. Please tell me if you have seen yesterday's class. You have. Good. There are some no's also. So please ensure that you go through the classes. And if you have any doubts with regard to even yesterday's class, we'll discuss it. Whenever I give you the time to ask your doubts, if you have some doubts regarding yesterday's class as well, you can ask it even in today's class. No issues about that. All right. But for those of you who are saying no and you've not gone through yesterday's class, please do that, because there are some very key aspects of biotechnology that we discussed yesterday. I would say that the most important topics related to biotechnology were actually discussed in yesterday's class, and there are few important topics that are left in today's class. So it's not that there will be any issue if you have not gone through yesterday's class yet, because the topics that we are going to discuss, they do not have any relevance from yesterday's class. So don't worry about that. But in case you've not gone through it, please ensure that you go through as quickly as possible. All right. So let's begin. The next topic that we have is DNA fingerprinting. Now, DNA fingerprinting as a medical application, we see that it becomes crucial. In the healthcare, there are ways in which DNA fingerprinting is used. Now, when it comes to DNA fingerprinting, understand why are we saying that this is DNA fingerprinting. All right. So if I ask you, what according to you is unique about fingerprint? Can you tell me what is unique about our fingerprint? What is unique about our fingerprint? What is unique about our fingerprint? The pattern, correct? No two of them are similar. Yes, this is correct, Sashwati. It itself is unique. Yes, that's true. So the unique thing about our fingerprints is that it is unique, that everybody has a different fingerprint. Even if we are talking about twins, even twins would have different fingerprints. So that's why when we say DNA fingerprinting, the same also applies to DNA, that no two people in the world will have the exact same sequences of DNA. There will be some differences in the DNA, but if there are people who are blood related, who are related to each other, for example, mother and daughter, father and daughter, mother and son, two brothers, sisters, etc., these people will have some similarity. There are some hereditary traits that will be seen between these two people who are related. So that's why there are some differences, yet there are some similarities, but no two people in the world will have the exact same set of DNA. Even if they are identical twins of the same gender, even then there will be some differences in their complete DNA pattern. So that's why this we see that this is something that is very, very crucial, that there will be some differences, yet if you are blood related, there will be some similarities that can help in recognizing the similarity, or rather the relation between two people. Now, DNA fingerprinting, now what we see is that it becomes very crucial in identification a lot of things. That we can actually sequence DNAs, we can identify DNA samples, and that helps us in various different ways. So, for example, application wise, the first one of the applications can be identification of criminals. So, for example, let's say there is a crime scene, and from the crime scene, there are certain DNA samples that have been collected by the police. Now, when they've collected these DNA samples, they can cross reference. Suppose that there is DNA sample that they found at the crime scene, and there are three suspects. All right, there are three suspects, and DNA sample of these three suspects have also been taken. So if you look at this particular picture right here on your screen, can you tell me which DNA sequence of out of these three criminals matches with the crime scene sample that has been collected by the police? Which one matches? Yes, the second one, correct. So if you see all the three sequences, this, and this, all the sequences are correct. All the sequences are matching, because in case of suspect 1 this doesn't match, this doesn't match. In suspect 3 this doesn't match, this doesn't match. So the only suspect where the crime sample matches with the DNA sample of the suspect is suspect number 2. So that's why it can give us some clues to the police that okay, this particular person was present at that crime scene. So identification of criminal. Similarly, we see that paternity tests are also done on the basis of DNA analysis. That DNA analysis will also help us establish certain relationships between parents and their children. Then even in unfortunate cases of accidents where, let's say, uh, identification of the dead bodies can't be done, there also DNA analysis helps in the identification of the dead bodies. So that is also where we see that it becomes crucial. Then in making drugs which are effective. Now, yes, day before yesterday, in the last, I mean, not day before yesterday, the first class of biotechnology, when we were discussing the medical applications of biotechnology, we had discussed one of the medical applications where we discussed how genes of a certain person would uh, respond to certain drugs. And there is a word that is used there, uh, the branch of, you can say, the branch of biotechnology that tries to identify or understand the genetic makeup versus how certain medicines would respond to certain genes. So there we had discussed that to make effective drugs we can make the use of it. Do you remember the term that we had used in the first class that we were discussing about biotechnology? This is where I'd given you one word about how genetic makeup is related to the medicines and the efficacy of medicines in a certain person. So that is something that becomes helpful in making drugs as well. Anyone who remembers the word? Not biopharmaceutics, one of the branches, or you can say one of the segments of biopharmaceutics. Yes, Shreya, you are correct. Anyone else who remembers? I see a couple of answers, but only one correct answer has been given yet. This was, yes, Satyapal, you are also correct. This was pharmacogenomics. Pharmacogenomics, where we are saying that how we are looking towards how genetic studies are related to pharmaceuticals, meaning that we can design medicines according to the genetic makeup of a person so that we can make more effective drugs. So that is what pharmacogenomics was about. So that pharmacogenomics basically becomes a part of how DNA fingerprint works. Then treatment of genetic vulnerability. Now this is very, very crucial because DNA fingerprint will help us identify certain genetic diseases. And with the identification of those genetic diseases, there are steps that can be taken in the offsprings. If, let's say, in a person we have been able to identify genes or identify the genetic diseases, then we can take certain steps so that can be saved from that disease. So that's why treatment of genetic vulnerability is also one of the applications of DNA fingerprinting. And then after that, also in wildlife management. Now, in wildlife management, also very crucially, it is something that can be useful. How exactly? Just think of it. DNA samples of animals can be collected and they can be studied. And that will also give us, especially when it comes to endangered or critically endangered animals or plants, even for that matter. Once we study the genes, their genes, we can actually understand what is leading to the downfall of their population, what's causing the downfall of the population. And that is something that can help us in taking certain conservative efforts as well. So those conservation efforts will help us in identifying the basic ways in which we can help in conserving their population. Printing becomes very, very crucial. So that's why there are multiple applications like these where DNA fingerprinting can be useful. But then there are concerns over DNA fingerprinting as well. So first thing, something that we had discussed before also, that it can lead to reference for designer babies. Now, discuss that designer babies is when we are trying to design offsprings with desirable properties. So when you are looking for certain characteristics that okay, my child should have this, my child should be like this, my child should be like that. So when you are doing that, when you are trying to find genes that will help in doing that, there is something which is called as germ line editing. Germ line editing. So this is something that leads way to germ line editing. Now, remember, when it comes to germ line editing can be both good and bad. Germline editing is something where you are making a permanent change in the genetic makeup of not only one individual, but for rather the generations or the upcoming generations as such. So that is called as germline editing. Now, germline editing is when we make the changes in the reproductive cells. We discussed about, if you remember, we discussed about cloning. And in cloning, we discussed that we use somatic cells. What are somatic cells? Somatic cells are the cells which are non-reproductive in nature. That is, any other cell in the body apart from the sperm cell in males and the egg cells in females. Apart from these two cells, every other cell in humans can be called as somatic cells. So in cloning, what we were doing is making the changes in the somatic cells. So when you make changes in the somatic cells, the change remains localized in that person only. All right. So it doesn't affect the future generations. But when you make germline editing, germline editing is when we make the modification of the genetic information in the reproductive cells. That is something which can also be passed on to the future generations. So once you make a change in these cells, they will be permanent in nature. So that's why you have to understand this. So germline editing basically is, you can say, modification of the genetic information in the reproductive cells, like for example the sperm cells or egg cells, etc. Sperm, egg, etc. that can be passed on to the future generations. So this is called as germline editing. Now this germline editing can be both good or bad. Now why am I saying this? That it can be good or it can be bad as well. So understand when this germline editing can be good. This can be good when we are trying to treating genetic diseases. Treating genetic disorders. right? So this is when it can be good, and also understand when it can be bad. So it can be bad when we are looking towards these kind of changes, like when we want to make cosmetic changes, or when we are looking at a concept like designer babies. So then it can be bad. So that's why germline editing can be both good and bad. Now in this case, when it comes to baking of designer babies, that you want certain characteristics. For example, let's say you want your child to be the greatest mathematician of the world. So you just have to find the mathematician right now, who is the greatest mathematician of the world, or probably the most successful one, and you would want those genes which are leading to that kind of aptitude. Similarly, you would want that I should have the greatest looking child ever. So you will find your best, or your favorite actor or actress, and you'll try to get those genes so that you can insert it in the embryos and try to make that permanent change. But when you make that permanent change, this is very bad and unfortunate and unethical at the same time, because you are making or taking the decision for somebody who doesn't know how to take decisions, and you have no consent of that child. So, for example, what if the child wanted to become the best lawyer in the world, but you wanted that the child should become the best mathematician in the world? And so it happened because those were the genes present in the child. So there was no permission taken, and there was no consent, and there was no realization in the child about what's happening. And yet you had taken that decision. So that's why this is going to also create a lot of problems. It can create a lot of issues altogether as well in the world. So that's why this can be both good and bad. So that is one issue. Similarly, genetic privacy of individuals can also be violated. Now, here what happens is that if, let's say, DNA fingerprinting is working everywhere, it also means that the information about the genetic makeup of an individual is also a public knowledge, right? Everybody knows about the genetic information of somebody. Now, what happens then? What happens then is another way of class divide, where you find ways to differentiate against somebody on the basis of genes. We as humans already have n number of ways in which we discriminate against somebody else, whether it is on the basis of race or language or money or religion or caste or whatever it is, world around, we have found ways in which we can differentiate against one person or the other. This will become a new way in which humanity will find ways to discriminate against. And that's why this genetic privacy being violated can be very, very dangerous. And then after that, it can also be misused for commercial purposes. How? Suppose that your genetic information is available with a pharmaceutical company, and now that pharmaceutical company knows if you have these set of genes, these could be the possible diseases that might infect you in the future, right? So once they know that, they always can play on your fears. There is no bigger fear than somebody's death. So if they start playing with that fear, they can create a great market for themselves without actually creating a lot of difference for their lives. Just that they would create panic, and they would start doing their business on that panic. So that's why it's all possible. And it's not that we are talking about a future science fiction. It's not that it might happen in the future. There are things that are happening today, where how science is being misused against you in a lot of ways. In a lot of ways, whether you go to one industry or the other, whether you go to the cosmetic industry or the food industry, or whatever industry you go, wherever science is being used, probably science is also being misused against you. You look at so many advertisements. Look at so many advertisements that you see on the television, and you see that how they claim these advertisements, they claim to be something else, but in reality they might be something else. And there have been certain cases in the recent past which are related to that, about false information being given through advertisements. For example, the very recent Nestle problem that we have seen, or the Patanjali problem before that, that we have seen in the past couple of months, where we have seen that the Supreme Court, uh, they have questioned these companies on the fact that, or saying that they are misleading the public into something, into believing something which probably is not true. So that's why these kind of things can happen, and they do happen. There is a wonderful book written on this kind of a topic, a wonderful book that I recommend in each and every class where I go. There's a book called as Bad Science. Now, Bad Science is a book about how science can be misused against you, because many of us, we don't understand science, and whatever will be told to you with a lot of vigor, a lot of excitement, uh, a very good script, then in that case there's a possibility that you would believe it. A lot of advertisements, you do believe it, and especially if they are trying to instill fear in your minds, you probably believe in those advertisements. So many a times that happens, and many a times these companies, they do not present the full truth, but unfortunately we do believe it because it sounds believable. Correct. uh, Very simple example. Charcoal. Today, if you go around, you'll find that most of your cosmetic products, they have charcoal. Your face wash has charcoal, your soap has charcoal, your shampoo has charcoal, moisturizers, I don't know what else. So everything has charcoal. Toothpaste has charcoal. Why charcoal? Probably nobody knows. Just that charcoal has become a thing and it's being sold in the market. So, and you go and you buy it because the advertisements seem believable, so you believe it. So if charcoal was so good, should we start eating charcoal for getting better results? Of course not, right? So that's where the problem lies, that you probably are using a charcoal based product as one of your cosmetic products at your home right now, but you don't know why. You just believed certain random advertisements that came across. So that's why this is what happens. I mean, I'm not saying that charcoal is bad. I'm just saying that you just believe it. Probably you don't know about it, but you just believe it. So there are all these problems, and that's why we know that science can be misused if you don't understand science. Correct. They will explain you anything and you will believe it. Right. What's the guarantee, for example, that everything that I'm saying is correct? I'm just joking. I'm just joking. Everything that I'm saying in the classroom is well researched and has been taken from multiple different sources. So it's not that I'm making up stuff. But I can if you don't understand science. So anyway, so now genetic information can also be misused to create weapons of mass destruction, right? Weapons of mass destruction can also be made, and we do know that this is possible. That how, for example, let's say, suppose Indian genes, there have been a lot of mixing among the Indian genes for thousands of years, because this is a part of the world which you can say has boundaries all over. You have the peninsular India, which is surrounded by seas, so not easy to cross the seas and come to India. Then you have the Himalayas, which is a natural barrier. You have the Karakorums, which is a natural barrier. You have the Thar Desert, which is a natural barrier. So that's why you have a lot of these natural barriers, which makes this subcontinent. Correct. Makes this part of the subcontinent. So obviously there have been a lot of genetic mixing in over 1,000 years. So obviously there are a lot of common genes which are found in India and maybe not elsewhere. Now, if one, let's say, any country wanted to lodge a biological attack against us, the very first thing that they have to do is to find those genes. Find the genes which are common in India and they're not found elsewhere in the world, right? Common in India and not found elsewhere in the world. So when that happens, this is where they can actually create weapons of mass destruction against Indians. So that's where the problem lies, and that is what we have to be aware of. So that's why all these things are actually possible and it actually can happen. So that's why DNA fingerprinting, there are a lot of concerns as well. Then after that, there is another topic, that is gene therapy, something that we discussed uh, day before yesterday. I mean, the first class of biotechnology as well, where we discussed that gene therapy is where we are making the use of genes, either by adding missing genes or by replacing certain genes, or maybe removing genes which are causing the problem, or maybe making certain modifications in the gene itself. So there are all these ways in which we can use gene therapy. So replacing a gene responsible for disease, inactivating or knocking out a gene, or introducing a new gene. There are these things that can be done in gene therapy. Now, when it comes to gene therapy, we discussed that there are a lot of problems that can be solved with the help of gene therapy, especially, let's say, genetic vulnerabilities or particular diseases where some genes are getting affected. So, those kind of areas is where we can take the help of gene therapy, and that gene therapy can actually help us in creating a scenario where gene therapy can solve problems which medicines, vaccines, or other medical therapies right now they're not able to solve. So that is where gene therapy can be useful. But what we see is that still there are concerns over gene therapy. There are three basic concerns that we see when it comes to gene therapy. The first concern is the short lived nature of the treatment. Why? The reason is that when you, let's say, are gene into the body, suppose that there was a missing gene, and that missing gene has been introduced into the body. What happens next? The next thing that happens is that once this gene has been introduced into the body, uh, it starts to function normally. But our growth and development mechanism sometimes does not let it function for very long. Understand what happens. There are millions and billions of cells in our body, right? And these cells, the cells grow, they develop into a fully developed or fully functional cells, and they die. And as they die, they are replaced with new cells. And this is a continuous process in our body. For example, by the time I finish one sentence, almost 1 million cells in our body would be replaced. So that's the speed. It's so quick that by the time we just finish one sentence, almost 10 lakh cells are getting replaced in the body. So that's a continuous process. And because of this process, sometimes what may happen is that the body goes back to the original way of working and forgets about the newly introduced gene or the modifications made in the gene. And that's why it is possible that the gene therapy is short lived in nature. So that's why somebody who's going through a gene therapy might have to undergo multiple iterations of gene therapy. So going through gene therapy only once may not suffice for them, and they might have to do that multiple number of times. So that's one of the issues with gene therapy. The second problem is our immune response itself. Now, our immune system has a very simple rule, and the rule is anything that is foreign is an enemy. So any foreign microbe, a bacteria or a virus, is an enemy. Any foreign chemical or a toxin is an enemy. And sometimes it doesn't recognize that the gene that is being introduced into the body, it's not an enemy. It would simply see that, look, this is foreign, so this is our enemy, kill it, destroy it. And sometimes that's why the newly introduced genes in the body, they might be killed or destroyed by our own immune system. So that also is one of the concerns that we have with gene therapy. So that's another problem. Then lastly, one of the issues is multiple or multi-gene disorders. Now, when we say multi-gene disorder, all right, understand this. Multi-gene disorder, for example, diseases like heart diseases or high blood pressure, Alzheimer's, arthritis, diabetes, these are diseases where not just one gene but multiple genes get affected. Now, when multiple genes get affected, it means that the modification needs to be made in multiple genes, and that's not going to be easy at all. That's not an easy task by any stretch of imagination. So that is where the problem lies. That in many of these cases, what we understand is that there are multi-gene disorders, and these multi-gene disorders are not easy to solve or treat with gene therapy. All right. Then after that, another problem that we see is that presently the technique remains risky. It has not proven to be very safe and effective in many of the cases. So that's why they do not respond very well. In some of the cases, they are short-lived. In some of the cases, they are not working. In some of the cases, they produce a side effect. So there are all these chances with gene therapy. So that's why we see that stability is something that we are still trying to attain. At the same time, one more problem is cost. Right now, it is costly. Right now, it is not in the reach of everyone. And that's why what we have seen is that recently there are efforts with the help of AIMS and Health Ministry. So AIMS and Health Ministry, they have come together in an effort to reduce the cost of gene therapy and other costly treatment methods. All right. So trying to reduce the cost of gene therapy and other costly treatment methods. So that's why we see that there are these kind of initiatives that are being taken so that this can be reduced. Otherwise, there are a lot of these treatments which are way too costly for somebody to even try and experiment with. So that's why there are these concerns that we have with gene therapy right now. Then there is one more concept, and this is the concept of three parents baby. Now, three parents baby is a concept where we are talking about three parent, and understand why are we talking about three parent. One thing that you should know is when it comes to the cells, there are different organelles in the cells, and you might know that there is one organelle called as mitochondria. Heard of it? Mitochondria. I even know what some of you are going to write in the chat. Already many of you have written it. Powerhouse of the cell, correct? Powerhouse of the cell. Why do we call it as powerhouse of the cell? Yeah, they provide energy, correct? They provide energy to the cell. This is their role. Why they are called as powerhouse? They are called as powerhouse of the cell because they provide energy. They provide energy to the cell. All right, they provide energy to the cell to perform various different functions. Whatever energy is required by the body, by our cells, that will be provided by mitochondria. Now, one very or rather I would say two very important things about mitochondria. Mitochondria also contains DNA. We discussed that nucleus contains DNA, right? But it's mitochondria also that contains DNA, although the properties of those DNA are very different. The DNA that we have inside the nucleus, they are unique in nature. But the DNA that are there inside the mitochondria, they are not unique. They are found in entire species race. So for example, just understand what I'm saying. My DNA inside my nucleus is different from the DNA that you have in your nucleus. All right, DNA inside my nucleus is different from DNA in your nucleus, but the mitochondria in my DNA is similar to the mitochondria, sorry, the DNA in my mitochondria is similar to the DNA in your mitochondria. Understand the difference. Our mitochondrial DNA are similar, our nuclear DNA are different. Unless there is any non-human entity who is also sitting in the classroom, then I can't guarantee that. But for all the humans who are sitting and watching this class, for them I'm saying this, that all the mitochondrial DNA that we all have, we share common mitochondrial DNA, whereas all our nuclear DNA would be somewhat different. So that is basically what we see with mitochondria. Now, one more thing, when it comes to mitochondria, all right, mitochondria gets transferred from the mother to the child. Father's mitochondria, they don't get transferred to the child. Very important. All right. So only mother's mitochondria get transferred to the child not fathers. All right. So this is what we see with mitochondria. This is what we understand with mitochondria, that it's only mother's mitochondria that gets transferred to the child. Father's mitochondria do not get transferred to the child. Now, in this scenario, let's say if there is some kind of disease that has developed in the mitochondria of mother, if mother's mitochondria are diseased, then the child also inherits the same mitochondria. Correct? The child also inherits the same mitochondria. Now, what happens in that circumstance? What happens in that circumstance? That the same disease that was affecting the mother might also affect the child as well, which is something that we won't want. Correct? So, this is where we have found a way to avoid this scenario by having a donor cell, a donor who could donate the mitochondria. So if you look at this particular picture, what do you see here? We have mother's mitochondria or mother's cell which have unhealthy mitochondria. So what we do is that we remove the nucleus from the mother's cell, and this is inserted inside the donor cell. So we take a donor egg cell, and from that this nucleus will be removed, and nucleus from the original mother will be inserted, and now the fertilization happens between the male and the female cells, leading to the birth of a new child. So what happens is that the unique characteristics of the original mother has been transferred to the child, because nucleus has been taken from the original mother, and from the donor nucleus was removed. So that's why all the specific characters of the donor does not get to the child. The child will not inherit any properties from the any unique properties from the donor. All the unique properties will only come from the original mother and the original father. That's it. So that's why, remember why exactly this particular scenario occurs and how do we solve this. Now, last year there is a big breakthrough. Babies got three parents explaining mitochondrial donation treatment. So now this particular scenario is where we are talking about the same concept of three parents baby. There are multiple names that we call. So for example, assisted reproductive technologies, mitochondrial manipulation or replacement, or 3% in vitro fertilization. So these kind of names, plus as you can see, mitochondrial donation treatment. So this is all related to the same concept of three parents baby. So what we have seen is that there is a birth of a baby where with biological parents DNA and mitochondria from a donor. So that is something that has been done last year, May 12th, 2023. So that is basically what we see here in case of three parents baby. So there was this question, this was asked in your 2017 paper. In the context of recent advancements in the reproductive technology, pro-nuclear transfer is used for. So pro-nuclear transfer basically is the same concept of three parent baby, where we are talking about development. So we have first is fertilization of egg in vitro by the donor sperm. So this is nothing but in vitro fertilization, correct, or surrogacy, the basic surrogacy. Genetic modification of sperm producing cells. Now this is also not correct. Now this can be said to be close to germline editing, correct. So this is something which is related to in vitro fertilization or surrogacy, right? This is like surrogacy. And this one, genetic modification, in sperm producing cells, these would be close to germline editing. Germline editing. Then development of stem cells into functional embryos. So that again, it's just the use of stem cell for stem cell therapy. So this is something which is closer to stem cell therapy, right? Or reproductive cloning. In a manner, you can say that this is like reproductive cloning. And then lastly, prevention of mitochondrial diseases in offspring. This is exactly what we are doing here. So that's why the correct answer here will be D. Then there is the concept of GM mustard. But before going to GM mustard, if you have any questions, please let me know. Let's discuss some of the questions. Any doubts if you have before moving on to the next topic? What are test tube babies? So test tube babies basically is nothing but the same concept of surrogacy, where in vitro fertilization is taking place. That is, fertilization is taking place inside, outside the body of mother, and then the embryonic development is happening inside mother's body. So that is surrogacy, or what we also call as in vitro fertilization, or what we call as the concept of test tube baby. So all these three terms, they have the same meaning. Could you explain gene therapy shortly? So gene therapy is nothing but the use of genes for treating any disease or any kind of disorder. So when, for example, there is any disease that has been recognized that it is happening because of some kind of issue with the gene, either there is a missing gene, so we have to find that gene and insert it inside the body, or maybe there is a gene which is actually causing the problem, so maybe we can remove that gene or silence that gene, or maybe there is a replacement of a certain gene that is required. So all these will amount to gene therapy, where we are making use of genes for a medical treatment. Show me the diagram again. The diagram. Yeah, this is the diagram. DNA fingerprinting. So DNA fingerprinting is nothing but the identification of certain genetic sequences so that we can establish the similarity or differences in those genetic sequences. So why DNA fingerprinting? Because DNA structure of each and every person is somewhat unique, and that's why. Please repeat. Mitochondrial donor topic and process. So just understand this, that if mother's mitochondria are infected or diseased, we need to replace those mitochondria. To replace those mitochondria, we get a donor, and donor's mitochondria will be taken. So donor's egg cell will be taken, nucleus will be removed from that, mother's or original mother's nucleus will be inserted inside the donor egg cell, and then fertilization would happen between the male sperm cell and the female egg cell, which has the nucleus from the original mother and the mitochondria from the donor. That's it. That is what this uh, mitochondrial donor topic is about, as you've asked. Germline editing. Germline editing, simple thing. Germline editing is that when we are making the use of uh, reproductive cells to make any changes or modifications in the genes. So that is called as germline editing. So germline editing can be permanent, and they can be transferred from one generation to another. So that is why germline editing many a times is not used, or even uh, prevented from using, has been banned in many of the countries. Mails, many of the emails that I received, if it was about some of the issues that you are receiving, that you face, I have forwarded it to the required team, and they might get back to you about this. Cloning and germline editing different? Yes, it is. That just understand the basic difference. That in cloning we are using somatic cells. We are using somatic cells, and somatic cells are non-reproductive in nature. Whereas in germline editing, we are deliberately using reproductive cells, because the changes that you make in the reproductive cells, they are permanent and they will be passed on from generations to generations. So that is the basic difference between the two. How gene therapy is related to drugs? I wouldn't say that they are related, because gene therapy is one of the medical treatment methods. Donor of mitochondrial DNA. I'm sure you wanted to ask mitochondrial DNA should be male or female, because you are taking the egg cell. It has to be a female cell. And anyway, father's mitochondria are not making any difference. So it doesn't matter. It's all about mother's mitochondria. So we'll have to find a female donor. We can reduce the sugar patients too by modifying the DNA. See, that is where we find that it becomes very difficult, because when it comes to genetic modification, or let's say even gene therapy. So, in case of gene therapy, what we see is that using gene therapy for multi gene disorders is not that easy. So, that is where the problem lies. All right. So, it's not going to be easy to use gene therapy in these kind of scenarios. IVF stands for in vitro fertilization. That is basically surrogacy, where fertilization is happening outside the body. What are the recent changes in surrogacy rules? Recent changes, mainly one of the recent change that we have seen in the surrogacy regulations rules of 2022, the changes that we have seen is that married couples are allowed to go through surrogacy, that they can use donor cells. This has been allowed to them, and this basically comes as a big relief to a lot of people who had complications, and because of complications they could not conceive and they could not bear a child. So now that those rules have been updated in India. Then there are a lot of repeated question. Germline editing has impact on physical appearance or cloning? It can have impact on physical appearance, because you are deciding the physical appearance many of the times. Vaccines are made with help of genes. Yes, of course there are vaccines, especially the nucleic acid vaccines, as we call them. There is DNA vaccine, there is the recombinant vaccine, vector vaccine. In all these cases we do make the use of genes. After introducing new specific genes, their qualities can be transferred to generation. Yes, that is exactly what happens in germline editing. How pharmacogenomics is used. So pharmacogenomics is all about understanding the genetic makeup of a person, and then make effective drugs according to that genetic makeup, whatever suits the best to that genetic makeup. So that's why it is very, very useful. Only female mitochondria transferred, not fathers. What if fathers mitochondria defect? That's the entire point, no? What has been written here, that only mother's mitochondria get transferred to the child, not fathers. So even if father's mitochondria are defective, they will remain localized in the father, they will not be transferred to the child at all. Is surrogacy same as IVF? Yes, it is. I'll write it here. See, surrogacy, IVF, the concept of test tube baby, they all mean the same thing. Difference between surrogacy and 3% technology. Very simple. Just understand this. In surrogacy, what are we doing? The fertilization is happening between the mother and the father, and but the fertilization happens outside the body because mother has some complications. So outside the body, or there will be somebody who will carry, and the embryonic development will happen inside. But in this particular scenario, we have to take mitochondria from the donor. That's not the case in surrogacy. Being infertile affects the social position of women. What's your view? It still happens in rural area. I mean, I wouldn't say not only rural India. It happens in urban parts of the country as well. And for anyone, I mean, anyone who is not able to conceive, who are infertile, it becomes a big challenge for them. Not only for women, yes, for women, of course, it becomes a much bigger problem, uh, much bigger issue. And yes, in the areas which are where the literacy rates are lower, where the understanding of different ways in which these problems can be solved is lower, we see that it does so happen that it leads to these kind of issues. If any health issue problem in, no, in three parents baby, not that we know of, it is something that can be done. Means if father have any disease, less chance of transferring to the child. See, not about any disease, about mitochondrial disease. If father has a mitochondrial disease, that wouldn't be transferred to the child. If there is a genetic disease that the father has, it can be transferred to the child. Ria, for the weekly test issue that you're facing, if you can drop an email, or if you have already written an email, send it to me. I'll talk to the people. Same Dhanya also about your history and polity related doubts, that whatever you have, or optional doubts, whatever you have. You, if you have already emailed or done something like that, it's fine. Otherwise, send an email to me. I'll get it done. Hmm. So let's move on to our next topic. And now this topic is GM mustard. Now, GM mustard very often has been in the news, especially since 2018. 2018 and 19, it was very much in the news. A question also was asked in your prelims examination. Then after that, in 2022, 23, again it has been back in news. So that's why first understand what is this GM mustard and why is it in the news. So first thing, this GM mustard is, GM stands for genetically modified. So it is a genetically modified mustard called as Dhara Mustard Hybrid 11 or DMH11. DMH11 or Dhara Mustard Hybrid 11. Now, what is this DMH11? DMH11 basically is a hybrid of mustard. Alright, it is a mustard hybrid. Now, this mustard hybrid has been created so that we can improve the productivity. We can improve the yield of mustard variants in India. And that's why in the tests, what we have found is that this genetically modified mustard gives us almost 25 to 30% better yield as compared to the best of the variants of mustard that are being grown around the country. So that's why this GM mustard is something that was first created, and it was created by the Center for Genetic Manipulation of Crop Plants in Delhi, in Delhi University. All right, it was created in Delhi University. So it was developed here, and then it was also passed on to the government. Then the Genetic Engineering Appraisal Committee, they also had a look. They also did variety of tests on it to also understand the problems, or if at all there are any problems related to it. And then they had cleared it for releasing it into the Indian market back in 2018. Then after that, the central government also gave its go ahead, and it was about to be launched into the Indian market. These seeds of GM mustard, they were about to be launched into the Indian markets, and this is when the Supreme Court intervened, and Supreme Court did not let the mustard seeds or the GM mustard seeds be released into the Indian markets. They did not allow for that. To understand why, what happened, that the government was okay and the government was about to launch it, and the Supreme Court intervened and did not let it release, and it held it back. And then it also formed a committee. The Supreme Court said that there should be a committee that should look after every problem related to GM mustard, and then come back with a report whether it is safe for releasing into the Indian markets or not. Now, what are these problems? What kind of problems are we talking about? The problems are twofold problems. One, if understand the issues raised against GM mustard. Issues raised against GM mustard. Understand these issues. The very first, if they could possibly cause health issues. Then second large issue was biodiversity degradation. Now understand these two problems, that these are the two major problems that were raised. The first problem, if they could possibly cause health issues. Why? Because these are genetically modified and we do not know what kind of long term effects they can have in the body. So this was one of the primary problems that was raised, that we have we have not done a lot of research into what could be the negative impact on the human body of this GM mustard. So that's why more studies should be done. We should be 100% sure that's not going to cause any kind of problem in our body, and then only it should be launched into the Indian markets. So this is a firm belief that many of the activists who had raised their voice against the release of GM mustard, this was one of the concerns that they had raised. The second concern was with biodiversity, which was raised by a lot of environmentalists as well. Why? Today, and as the data was also reported, and the data as it suggested, that there are 66 variants of mustard in India. 66 variants of mustard, and they are being grown in various parts of the country. Once this GM mustard is ready to be released and the farmers could use it, then in that case, what might happen is that farmers will stop cultivating every other variant of mustard, and they might completely switch to this GM mustard. Then what happens is that over a period of time, many of these mustard variants, because they are not being used anymore, they start to decline. Their seeds will be destroyed and their populations will diminish, and it can lead to the extinction of certain these variants like these. So that's why it can lead to decrease in biodiversity of specifically mustard. The genetic diversity of mustard could decrease because of GM mustard. And tomorrow, what if there is a disease that is affecting GM mustard? It can just wipe out mustard variants because we are not using a huge variety or a number of variants of mustard wherever we are growing it. So that's why these are some of the issues that were raised with GM mustard. Now, recently, what we have seen is that there have been news like this. So, for example, a GM crop decision that cuts the mustard, And second is GM mustard DMH11's field trial likely to be delayed as sowing winter closes. So what we saw was that last year the Genetic Engineering Appraisal Committee again cleared the release of GM mustard. The expert committee that was set up, they have submitted their report, and their report is saying that we could not find anything that is alarming, so we cannot conclude that it might lead to any kind of problem. All right. So they are saying that they don't believe that it could lead to any kind of problem. So now, first the committee said that we are fine with it. Then the Genetic Engineering Appraisal Committee said that we are also fine with it. Now the government has gone to the Supreme Court, and there they have asked for permission, that you look at the report has cleared everything, give us a date, when can we go ahead with the release of GM mustard. The Supreme Court yet hasn't said anything as yet, and we have to see when exactly it happens. We could expect, because winter months is when mustard is grown, so we should expect that before 2024 winters, probably a decision will be taken by the Supreme Court. So until then, this is where we are. Now also understand that why the government is also so much adamant on releasing GM mustard. Why do they want that this GM mustard should be available for the farmers? Now, mustard, as you all know, that mustard is used everywhere in the country, correct? In one form or the other. Whether these are mustard seeds or whether it is the mustard oil, in one way or the other, we see that mustard is being used or being consumed everywhere in the country, right? But almost 60% of the mustard that we need in India, 60% of the mustard is imported. Productivity of mustard in India is not that great. And that is where the government is very keen, because it thinks that with the release of GM mustard, we would be able to increase the efficiency, we'll be able to get better product, produce better yield, and that would decrease our import dependency with regard to mustard. And that's why it might lead to cutting down the cost of mustard or mustard oil. So that is where they are. That is what the thought process for the government is right now. So that's why with GM mustard, they expect better productivity. They expect better productivity. All right. With GM mustard, they expect better productivity, and that's why uh, it might lead to. And it's a big part of everybody's food in India, and that's why they are concerned about this. So that's why we see that there are all these things with regard to mustard, or rather GM mustard. This question was asked in your 2019 paper. It's a fairly simple question. There's one statement that does the work for you. I'm launching the poll. Please mark your answers. It's a very, very easy question. Good. 66% of you have marked B, which is the correct answer. Genetically modified mustard or GM mustard developed in India. Consider the following statements. Look at the third statement. Forget about the first and second statement. We did not discuss anything about first or the second statements. Look at the third. GM mustard has been developed jointly by IARI and Punjab Agricultural University. Has it been done or developed at Punjab Agricultural University? The answer is no. It has been done where? Center for Genetic Manipulation of Crop Plants in Delhi University. So that's why it's a simply a wrong statement. You can say that, because when it comes to statement number 3, it alone solves our problem. Three out of these four statements, they have statement number three given in them. So obviously all these options are wrong. So correct answer will be B, that is true only. So even though we have not studied anything, we have not discussed anything about statement number one and statement number two, yet we can get to the correct answer only on the basis of statement number three, because that is something that we know, that the development is something that happened at Delhi University and not at Punjab Agricultural University. So that's why we get the answer. So now looking at statement number one and two. First statement says GM mustard has the genes of a soil bacterium that give the plant the property of pest resistant to a wide variety of pests. So what, see, this is basically not about pest resistance. We are trying to improve the productivity. We want to enhance the yield altogether, but not about pest. It's not about pest. That is something that we discussed in case of BT cotton. So that's why this is a wrong statement. Second statement is GM mustard has the genes that allow the plant cross pollination and hybridization. This is correct. Although we did not discuss this, but this is correct. So that's why just understand that when it comes to this is how the things are. Now, coming to the last topic of our discussion in terms of biotechnology, and this is about dark biotechnology. Now, dark biotechnology is where we are talking about the misuse of biotechnology. That's why dark. This is about misuse. Misuse of biotechnology. Misuse of biotechnology. So that's why when it comes to misuse of biotechnology, there are a lot of ways in which biotechnology perhaps can be used in a negative sense, or it can be misused, and that is what is a lot concerning. So there are various different ways. For example, bioterrorism, biowarfare, biocrimes, anti-crop warfare, and there are other things that we'll discuss. So for example, when it comes to bioterrorism, bioterrorism is when we are making the use of any biological agent like microorganisms or pathogens to cause problems, where for example, let's say uh, a certain disease is deliberately being released into the populations. That is what? That is bioterrorism. A certain disease is deliberately being released on the plants so that crops can be destroyed. Even that amounts to bioterrorism. So that's why all these are ways in which bioterrorism can be defined. Let's, for example, take the situation of COVID 19. There are various different reports that have said that COVID 19 may not have been naturally released into the atmosphere, but perhaps it could have been prepared in a lab. The virus might have been developed in a lab somewhere in China, maybe in Wuhan. We do not know that for sure, but we know that there are reports that say that claim this. Even there are US backed reports that claim this. Whether this is correct or not, we don't know. But we know that this is doable, and we have seen what kind of devastating effect it can have on populations across the globe. So that's why that is something that amounts to bioterrorism, if it is something that is being done deliberately. Similarly, it is not only about lives, that somebody could lead to somebody losing their lives, but even somebody trying to hurt the economy. Suppose a country like India, which is hugely dependent on agriculture. Let's say tomorrow some country is releasing certain pathogens in a manner that it is going to affect the crops and the productivity of the crops. And if it affects the productivity of the crops, it can have devastating effects on India's agriculture and hence India's economy. So that also amounts to bioterrorism. So that's why there are multiple ways in which bioterrorism can be used. Similarly, we can regenerate extinct pathogens. Last year, this has been in the news and fairly discussed last year. Scientists revive zombie virus which spent 500 years in frozen Arctic. Now this is something which can be very dangerous, because we are at least exposed to a lot of different types of viruses. A virus that has been dormant, that has been hidden for almost 48,000 years, if at all by accident it is unleashed on human population and it can actually affect human population, then we do not have anything, we have no answers, we have no vaccines, and it can cause another pandemic kind of a situation. So that's why these kind of things can be dangerous. Similarly, some of the pathogens that have been eradicated, let's say smallpox, plague, these kind of pathogens that cause these diseases, they have been eradicated with wide vaccinations all across the world. But today vaccinations don't happen. For the future generations, they do not have that natural ability to act or fight against smallpox or plague. They do not have that immune system which knows how to do that. So today if it is unleashed on population, we do not know what will happen. So that's why all these are also negative aspects of biotechnology. Then the effect that it can have on biodiversity. So what we know is partially something that we discussed in context of GM mustard as well. We know that when it comes to biodiversity, or when it comes to biotechnology or the use of biotechnology, there are only certain species about which we are concerned, right? We're not really concerned about a lot of different species. We are concerned about only a few, right? We are concerned only about a few. Now, in this particular scenario, because we are concerned only about a few species, we take all the precautions, we take all the steps also only to conserve these species, correct? We want only these species to do better. So out of, let's say, 100 species or 100% of the species that right now reside in this world, 99.9% of the species perhaps they do not get anything from us, or they have no value for us, so called value for us, so we do not do anything about them, and that can affect biodiversity, because certain plants or certain animals only will be looked after and others will not be looked after, so their population will start to decrease and it leads to decrease in biodiversity, loss of biodiversity. So that's why this is another problem. So that's why we have to understand that how it can have an effect on biodiversity. Then there is another concept of terminator genes. Now, terminator genes is something that is used in developing a lot of seeds of various plants. Where if a terminator gene has been inserted inside a certain seed or certain plant, uh, then that seed germinates only once. So, for example, let's say as a farmer, you go to the market, you buy seeds of a certain plant. And once you sow the seeds, you see that you get very good results. Right? You see very good results. So, the next thing that you try to do is that you want to conserve these seeds, right? You would want to conserve the seeds so that the same seeds can be used in the next season, because you have seen such a good productivity and maybe also with a very good quality, that both in terms of quality and quantity you have seen something extraordinary. So you would obviously want to conserve those seeds so that you can use the same seeds again the next year. But in the next year, when you're using these seeds, you see that the seeds don't germinate at all. Why? Because the company from which you bought these seeds, they had used terminator genes. And whenever terminator genes are used, the plants can actually uh, plants or other seeds can germinate only once. So that is where the problem lies. So terminator gene basically stops the plant from releasing fertile seeds. All right, it stops the plant from releasing fertile seeds. So that's why the farmer again is required to purchase the seeds in the next cropping season. And by that time, the company might already have increased the prices or made the farmers dependent, because last time you saw a very good yield, so you want to repeat that and you will want to go back to the same company. So then in that case, the company starts to make better profits as compared to others, which is an unethical practice. So that's why this is what the problem with terminator genes can be. Not only that, it's not only the effect that terminator gene can have only on this crop, but there can be instances of cross pollination with other crops which are growing in the same field, and the terminator gene might be passed on to other crops as well. So that's why it may cross pollinate with the local varieties and may affect the continuity of agriculture altogether. So that's why there are all these concerns that we have when it comes to terminator genes. So this is about all the negative aspects of biotechnology. Now, lastly, before we end this chapter, also let's have a look at the questions that have been asked in the mains examination from this chapter. The questions that they have asked, you'll see that the questions have been fairly simple in their nature. It's not very difficult to write answers in them. So if you look at the first question, 2021, what are the research and developmental achievements in applied biotechnology? Second question, how will these achievements help to uplift the poorer sections of society? So first, research and developmental achievements in applied biotechnology. Now, I'm sure that you do not know what is applied biotechnology, but you can guess. You can guess what should be applied biotechnology. Wherever we are making the use of biotechnology for something practical, in whatever field it is, that should be applied biotechnology. So what are the research and developmental achievements? Now there are a lot of achievements. There are so many examples that we have seen all throughout the chapter, which can be written here. The second is, how will these achievements help to uplift the poorer sections of the society? So then you have to write, you have to go one step further and also keep writing, right? Also keep writing about each and every example and the relation between the first and the second parts of the question. It's a very simple question based on probably the entire chapter, and you'll have to summarize this entire chapter in trying to get your answer. Then the second one is, how can biotechnology help improve the living standards of farmers? Now, this is nothing but, you can say, all the agricultural, all the agricultural applications that we have studied, all the agricultural applications that we have studied, just that you have to change your language, because it is not asking you how does biotechnology help in agriculture, or what do you mean by green biotechnology and how does it help. So you don't have to, that's why you cannot write directly all those points that we discussed, but related to the farmer and right from a farmer's perspective. That is how you'll have to change those points. Otherwise, these are going to be the same points that we have discussed. Then the third question, what why is there so much activity in the field of biotechnology in our country? How has this activity benefited the field of biopharma? Now, why so much activity in the field of biotechnology? Of course, a lot of activities, because there are so many sectors that we have identified. There are so many ways that we have identified where biotechnology can be very, very crucial, and it can lead to a lot of improvements in various different sectors. So that's why it's so many activities. And then after that, how has this activity benefited the field of biopharma? So then you simply have to write about biopharmaceutics and all these things that we discussed about biopharmaceutics in this question. And then this last question, this was asked in 2017. Stem cell therapy is gaining popularity in India due to treat a wide variety of medical conditions, including leukemia, thalassemia, damaged cornea, and severe burns. Describe briefly what stem cell therapy is and what advantages it has over other treatments. Now, if you notice this, the first part of the question is not really a question, but a sentence that is just appreciating stem cell therapy. Correct? So, it is fairly, you can say, a direct and a positive question where you do not really have to write about the negatives. So describe briefly what is stem cell therapy and what advantages it has. So you first have to define stem cell therapy, write certain few things about stem cell therapy before going on to all the advantages that stem cell therapy has. We had discussed 4 or 5 different topics where we discussed stem cell therapy. So this is what you have to write. In most of the questions, when it comes to, especially when it comes to biotechnology, you'll see that the questions in the mains examination are fairly simple in their nature and very static, the way they ask you questions. So any question if you have related to the last part of our discussion in last half an hour, please explain question number 1. So question number 1 says, let me just go back. that what are the research and developmental achievements in applied biotechnology. So write about the various advancements that have happened due to biotechnology. So everything that we discussed in agriculture, everything that we discussed in industries, everything that we discussed in green biotechnology, or rather red biotechnology, or medical field, environment, you just have to write all that. So that is basically what you have to write in advantages, or let's say achievements of research and development in applied biotechnology. Now, how does it help to uplift the poorer sections of the society? It helps to uplift the poorer sections of the society because it helps with a lot of things. For example, let's say agriculture. It has helped in increasing the productivity. It has helped in growing crops where traditionally we could not. So in all these cases, it raises the farm income. Similarly, we have seen that in medical field, it has been useful in bringing down the cost of treatment, in bringing down the cost of medicines, of vaccines. So all these are the advantages. In industries also, biofuels have come in. So advantage when it comes to or creating new opportunities with biofuels. So there are a lot of these things that can be written here. Question number four. So question number four basically is about what? It is about stem cell therapy, and you have to just write about the advantages of stem cell therapy and why this has become such an important treatment. And the advantages, one of the most important advantages, that it is something that can solve the unsolvable problems. Whatever other treatments have not been able to do, it can do. So that's why it becomes crucial. Why half an hour? I don't understand your question. Also, a quick announcement for all of you today onwards, and this is relevant for every one of you who is also attempting the paper in 2024. If you have filled the form for 2024, today onwards, we are also going to begin with the target prelims sessions, where we'll all our teachers and all of us will come up with the most important topics of the last 1 year, which would be important for your prelims examination. So today onwards, that is something that is starting. So the classes would start from 7 o'clock, but they will be available on the channel. So even after the class, you can go back, or you can just go through the questions and the discussions that we are going to do in these sessions. And all of that is going to happen on the YouTube channel. You are saying only half an hour left. I did not say that. I told you that whatever we have discussed in the last half an hour, 6 o'clock to 6:30, you ask those questions, because before that we discuss everything. I've sent you two mails. I'll check if, let's say, if it is unanswered, because I see the problem also is that in a day I actually sometimes receive over 70 80 odd emails, and I try to respond to every one of them or forward it to the required teams which have to solve your problems. But if they have been left unanswered for some reason, you just resend those mails to me. These topics enough for Karnataka PCS? I wouldn't say that. See, the way Karnataka PCS will ask you questions will be little different, because they will be more factual in nature as compared to the civil services paper. So that is the basic difference. So yes, these topics are helpful, but the way we would discuss perhaps will not be as useful. You have to also do one extra work, and that is also gather the facts about these current affairs. Glaciers are melting. Venezuela has exhausted what India should do. See, this is going to be a big challenge, because we see similar issues also happening in India. This year, you might have seen that there has been a lack of snowfall, especially in January in the northern parts, in Uttarakhand, in Himachal, in Kashmir, in all these areas. We saw that the snowfall was not adequate enough, and that is something that has led to severe water stress even in India. So the only way to reverse it is to reverse climate change, is to stop or at least reduce the carbon emissions. That's the only way to go about this. Same website or YouTube channel? Yeah, the same channel. There is only one Byju's IAS channel, that there only all the classes will happen. Is this enough for Karnataka KS? Look, uh, Karnataka KS examinations are uh, factual. I mean, there are many of the questions which are factual, and there will be one step down of how the civil services paper would be. Yes, it does help. I'm not saying it does not help. It will help you, I would say, for 60 to 70% of the syllabus. But for the rest 30% of the syllabus, you need some current affairs facts. You also need some general science, because general science questions are not asked in civil services exam, but in KS exams it will be asked. So that is one change that you have to do in your preparation. Recently we experienced northern lights. Can we expect questions from that particular topic? Yes, we can expect questions from there, but although it could be a bit late, and I'm also expecting that the question papers already would have been created before these things happened this year, but it actually also happened last year. So that's why they could take a reference. For example, this year, you if you know, on 11th of May in Ladakh, aurora was seen. Northern lights were actually observed in Ladakh in on 11th of May, just a couple of days back. But last year also it happened. Last year in April also it happened. So it's possible that they can ask this question, not referencing it this year, but rather the last year. So it's 6:33. So let's take a break. Let's be back by 6:45. Let's be back by 6:45 and continue with our discussion. I will start with our next chapter, that is nanotechnology. Welcome back, everyone. Now, the next chapter that we have is nanotechnology. Now, nanotechnology as a chapter, again, I would say that this is a chapter which is more important for your prelims examination as compared to your mains examination. And there are some basics of nanotechnology that we have to discuss. Now, when it comes to nanotechnology, especially when we look at the pattern, you will see that in the past 5 6 years, the number of questions from nanotechnology has increased, or rather, I would say 6 7 years, let's say 2017 18 onwards, the number of questions actually have increased compared to the kinds of questions or the number of questions that were asked from nanotechnology in the past. So that's why I would say that from prelims perspective, especially, I would consider this to be an important chapter. And also, any new discussion or any new current affairs also becomes important parallelly. Now, first and foremost, what do we mean by nanotechnology? Again, if you just try to break down the words, you have two words here: nano and technology. Now, nano, the word nano basically has a meaning in Latin, that is dwarf. So nano in Latin means dwarf. But then there is this name also given because of the size of 1 nanometer. Can you tell me how much is 1 nanometer in terms of meter? How much is 1 nanometer? Yes, 10 to the power of minus 9 meters. 1 nanometer is 10 to the power of minus 9 meters. Now, when it comes to nanotechnology, one way we can say that this is technology of the dwarf or technology of the tiny or the very small particles. But essentially, we are talking about technology or science related to the particles which are of the order of some nanometers: 1 nanometer, 10 nanometers, 100 nanometers, or maybe zero point 0.1 nanometers. So this size range of, mostly you can say, 1 to 100 nanometers is where we have nanotechnology. The technology related to the study of particles which are 1 to 100 nanometers in size can be called as nanotechnology. So that's why nano term comes from the Latin word for dwarf, and in scientific terms, it refers to nanometer or 1 nanometer. Now, in case of this scale becomes very, very crucial. Just understand this. The term nanotechnology was coined by a Japanese scientist, Norio Taniguchi. Now, understand why 1 nanometer and what is so special about this scale of nanometer, that we have an entire branch of science and technology that has been named after this. There is micrometer also, there is millimeter also, but there is no milli technology, there is no micro technology, but there is nanotechnology, correct? We are not studying any micro or mini technology, we are only studying nanotechnology. So what's so special about this one segment or this one scale, that is nanotechnology? So understand this. This size of 1 nanometer and thereabouts is the size where the molecules start existing. Molecules of different substances, they start existing at this scale of 1 nanometer, or you can say range of 1 to 100, or maybe zero point 0.1 to 100 nanometers. Now, why this is important that molecules are formed here? Because molecules, they have very specific properties, and the materials that you are forming, they are formed out of these molecules. So for example, let's say before or below molecules you have atoms, right? You have atoms. Now these atoms, what's special about atoms? Atoms of, let's say, oxygen will always remain atom of oxygen wherever they are. But when they combine with something, or when they start forming a molecule, this is when they start adopting a certain property. Let me just give you an example. You have hydrogen atom and you have oxygen atom, right? Hydrogen and oxygen atom. When we combine these two, there are two ways in which they can be combined. either as H2O or as H2O2. Either as H2O or as H2O2. So either as water or as hydrogen peroxide. Now, what's the difference between the two? Of just one oxygen, that H2O has one oxygen and two hydrogen, and H2O2 has two hydrogen and two oxygen also attached to it. That's the only difference, just one extra oxygen. But this one extra oxygen completely changes the properties of these two substances. Water, I need not tell you the properties of water. I need not tell you about the use of water. You drink water. I mean, I have this water bottle right here in front of you, which I keep sipping from time to time. So you and we know how essential water is for life sustaining on this earth, correct? On the other hand, you have H2O2, that is hydrogen peroxide. And hydrogen peroxide, first of all, is not drinkable. Please don't try at home. Secondly, it is an industrial reagent. It can be used as an oxidizing agent. It can be used as an antiseptic. So there are a lot of different ways in which H2O2 or hydrogen peroxide can be used, but those are not the ways in which we use water. Those are not applications of water. So there are stark differences between uh, H2O2 and H2O just because of one extra oxygen. And these are molecules. This hydrogen as hydrogen atom would always have the same property. Oxygen as oxygen atom will always have the same property. But when it combines in various different ratios, we see that there are different properties that come up just because of how they combine. And forget about hydrogen and oxygen combining. Take example of carbon. The same carbon in its form can be coal, about which we discussed that it becomes a fuel. The same carbon also becomes graphite. The same carbon also becomes diamond. Diamond is a precious stone, right? Very costly. And what is graphite? You use it in pencils. So graphite also is a pure form of carbon. Diamond also is a pure form of carbon. What's the difference between these two? Their structure, the arrangement of carbon atoms. So only because of the way carbon atoms have arranged among themselves differently, there are two completely different properties that we see in these two substances. Where is all this happening? At the nanoscale. Where do you start seeing all these properties? At the nanoscale. And if you had to manipulate certain properties, if you had to change or modify certain properties, where would you do that? At this nanoscale. So that is the importance of nanoscale. And that is why there is this entire branch of science which has been named as nanoscience or nanotechnology and so on. So that's the importance. Let me give you just another example, a hypothetical example. Suppose that there are two surfaces, all right? There is this one surface which is like this, and there is this one surface which is like this. And let me ask you a question here. If I ask you which of the two, let's say I'm calling this as A and I'm calling this as B, which of the two would have a higher melting point? If they were made of the same substance, which of the two would have a higher melting point? I'm starting a poll for you quickly. Which of the two will have a higher melting point? A or B? Interesting. That 55% of you feel that it should be A and 45% of you feel that it should be B. Can you tell me your reason? Can you tell me why you feel that A should have higher melting point or why you feel that B should have a higher melting point? What is your rationale? Also tell me, I mean, with the answer, I mean, what was your answer and what is the explanation? Otherwise, I would not know what you mean. Yes, Nikhil, you've given the correct answer. Satyapal, you've given the correct answer. NB, you have given the correct answer. Okay, Samiksha, you have given the correct answer. All right. Vivek, right? Correct. So let me tell you a very simple rationale about how we are thinking. Well, let's say when you apply heat, suppose that you are applying heat to A and you are applying heat to B. All right. So when you are applying heat, what happens? All the heat in A gets concentrated at one point here. But in B, first there will be a distribution of heat. First, there will be a distribution of heat in the entire surface, and then it will start melting. So, which will melt first? A will melt first. So, which will have a higher melting point? B would have a higher melting point. A would melt first because all the heat will be concentrated at one point. So it will be way faster in melting if they are made of the same structure or the same substances. Whereas in case of B, it's a wider surface. So first, all the heat gets distributed in this entire surface, and then after that, it starts melting. So that's why B would have a higher melting point. Now, let me tell you why when I did I ask this question. At nanoscale, we know or we are trying to find ways to understand how to manipulate all these surfaces or manipulate the properties of substances. And one of the ways in which we can manipulate the properties of substances is this. Suppose that we wanted to raise the melting point of a certain substance because of some application, we wanted higher melting point. What do? We change the structure. Is it possible? Hypothetically, yes, it is possible. Have we done it? No. But is it possible? Yes, it is possible. So that's why we can actually do that. And that is where nanotechnology is going to play a big role. That there are a lot of changes, manipulations, property modifications that can be done when you understand nanotechnology well. So that's why we see that there are a lot of researches that are happening today in the field of nanotechnology just to understand how exactly the things are. Now, in this particular scenario, when it comes to nanotechnology, we say that it is a branch of science that deals with the minute particles in the range of 1 to 100 nanometers in size. 1 to 100 nanometers in size. So now in this particular scenario, when it comes to nanotechnology, we understand that there are a lot of different things that can be done at the scale of nanotechnology. And overall, when you look at this scale, so for example, this part or this part of scale, this you can say that this is the nanoscale. This we can say is the nanoscale. Now, when it comes to nanoscale, this nanoscale becomes useful because this is where the as you can compare to everything that is bigger than this. That even when you compare to the red blood cell, the red blood cells or our blood cells, they are, you can say, 1,000 times bigger in size as compared to the nanoparticles. So that's why we can actually insert a nanoparticle in the bloodstream and make it go from one place to another. This is how actually nano drug delivery works. That we can do a targeted drug delivery with the help of nanotechnology by inserting the required medicine inside the bloodstream. So even that's possible. Even that's doable in today's world. So that's where we see that nanoscale becomes useful. And overall, when we look at our understanding of nanoscale, nanotechnology, understand this, that nanotechnology compared to many of the other technologies or other sciences that we have discussed until now, nanotechnology is a fairly new science. It's a very new science. The development of nanotechnology has happened only in the past 3 4 decades. That's it. We discussed about space technology, where you'll see that the observations have been happening for more than 1,000 years. We discussed about the energy sector, and we know that at least for more than 100 to 150 years, at least, we have been using the alternate energy sources as well. Nuclear technology more than 100 years now, 125 years almost. Biotechnology almost 100 years now. So at least since 1950s, enormous jump. But before that also, biotechnology was in use. But compared to that, when we look at nanotechnology, it's very new. Even the name nanotechnology did not exist until 1974, which was given by the name that was given by a Japanese scientist, Norio Taniguchi. Now, in this particular scenario, the development, or you can say the early development of interest in this field, starts with a very, very important lecture called as There's Plenty of Room at the Bottom. There was this lecture that was given by an American physicist, Richard Feynman. Now, Richard Feynman has been one of the most popular scientists, you can say, of the last 50, 70 odd years. A person, a scientist who was very well followed by other scientists, by students, by people outside the scope of the science world. He was very well followed, and he had become very popular. And that's why when he said something, it used to have a weightage, it used to have meaning, and it was being followed by many around the world. So he delivered a lecture in 1959 called as Plenty of Room at the Bottom, where he discussed that whatever we are doing with physics and chemistry right now, it's just at the surface. We have not tried looking beneath, and we do not even know what lies beneath. We do not have such powerful microscopes that can see at that level. So that's why we don't even bother, and we are not bothered about seeing at that scale. But what lies beneath probably will change the world. So this was, you can say, the direction in which this entire lecture went. And Richard Feynman has been very, very popular. There is a book also written on his biography called as Surely You Are Joking, Mr. Feynman. And Surely You Are Joking, Mr. Feynman is a wonderful read, first of all, and also one of the best sellers as far as any scientist's biography is concerned. Otherwise, these kind of books do not sell. A biography of a scientist, who reads, unfortunately? But his biography is one of the most read, followed biographies ever that we have seen. So that's why he was a very important scientist, and that's why whatever he said had weightage. And then we see that after this lecture, slowly and steadily, this starts to grow. A lot of interest is generated in the minds of the scientists, the engineers, and the students alike, and hence we see that a lot of growth happens. 1974, the naming happens, but everything changes in 1981, where scanning tunneling microscope was invented. Scanning tunneling microscope. The importance of scanning tunneling microscope, that with this microscope, now for the first time, we have been enabled to see at the nanoscale. Until now, the microscopes that we have, they were not able to go beyond micro level, or let's say 10 to the power of -6, about that scale. So in this particular scenario, we see that this nanoscale, this microscope, that is the scanning tunneling microscope, is able to see at the nanoscale. And this was very, very important, because now, until now, every study that we were doing was a theoretical study. But with the invention of scanning tunneling microscope, we now could see at this level of nanoscale, and this is a game changer as far as nanotechnology is concerned. So very, very important. And just understand how it works. There are two principles on which it works. One principle is called as tunneling, and the other principle is called as piezoelectric effect. So these are two principles on which scanning tunneling microscope is based upon. Now, these two principles, the first is tunneling. Understand what is tunneling. So when it comes to the scanning tunneling microscope, this is how the setup is. That there will be a sharp metal tip which is incident on the material. So it is touching the material, and it is pressing on the material. All right, it presses on the material. Now, as it presses on the material, electricity is produced. Now understand what happens. Let's say, let's say that this is the material. All right, this is the material that we are trying to study. Now what happens is that that here electrons are able to enter inside the surface like this. They will not be able to pass through completely, but they'll be able to enter inside by making a hole or making a tunnel for themselves. So that's why this is called as tunneling, because the electrons are able to make a tunnel through this particular material. And by making this tunnel through the material, they are able to show what's there in that material. So that's why they can see the material. The electrons help us in seeing that material, observing that material with the help of tunneling. So one principle that works here is the quantum mechanical effect of tunneling. So tunneling is the fact that electrons make a tunnel through the material, thus allowing us to see or observe the material. So this is what happens with tunneling, that the electrons are able to make a tunnel. The wave nature of electron is what we use here, that they would be passing on as wave and they would be able to enter inside the surface. The second is the concept of piezoelectric effect. Now what is piezoelectric effect? Piezoelectric effect is an effect where we see that electricity is generated. This is when you generate electricity by applying mechanical stress or pressure. This is what we call as piezoelectric effect. All right, this is what we call as piezoelectric effect. So piezoelectric effect is, so suppose that you have a solid surface or you have a solid crystal surface and you press this. If you press this and by your pressing, if electricity generated meaning electrons being produced. So if electrons are produced when something is putting a pressure or applying mechanical stress, that is called as piezoelectric effect. And that's what we use here, that the metal tip will be touching the surface, and not only touching the surface, applying some pressure on it, applying some stress on it. And as they apply this stress, this is where electrons will be generated, and these electrons, they are able to pass through this material, that is tunneling. So that's why piezoelectric effect and tunneling, these two become the primary principles on which the scanning tunneling microscope is based upon, right? So now piezoelectric effect also, there was one very peculiar news last year. Until now we have observed piezoelectric effect only in solids. All right, until now we have only have seen them in solids, but for the first time last year, scientists were able to observe piezoelectric effect even in liquids. Even in a liquid, for the very first time in the history of science, this has happened, that they were able to observe piezoelectric effect even in liquids. For the first time it has happened. So this is how scanning tunneling microscope works. Now there was another microscope, and this is atomic force microscope. Now a very interesting thing is that every topic perhaps that we are going to discuss one after the other, every topic has a Nobel Prize. Every topic got a Nobel Prize. So for example, scanning tunneling microscope and atomic force microscope, both were given a Nobel Prize, because this was a big invention, changes the world of nanotechnology. So both of them together were given the were given the Nobel Prize in chemistry. Hmm. So atomic force microscope. Now there is one limitation that the scanning tunneling microscope has. We discussed that there is a metal tip, electrons are being produced and electrons enter the surface. Now electrons entering the surface is only possible with conductors, right? If you remember the concept of conductors and insulators, insulators are the ones that will not let electricity pass through them, and conductors are the ones which can allow electricity to pass through them. So in the non-conductors or in insulators, passing or passage of electricity is not possible, rather passage of electrons is not possible. So that means that scanning tunneling microscope cannot be used there, right? Scanning tunneling microscope can't be used there, can't be used in cases where you have a an insulator or a non-conducting surface. So that's why we see that it becomes very, very crucial, because the limitation of scanning tunneling microscope is overcome with the help of with the help of atomic force microscope. So that's why what we see is that here it just goes, I mean, touches the surface, goes across the surface, and creates, you can say, a three-dimensional map of the surface to understand the properties of this material. So, so this atomic force microscope overcomes the limitation of scanning tunneling microscope, as scanning tunneling microscope is useful for conductors, whereas atomic force microscope can be useful for insulators too. All right. So that's why atomic force microscope was an improvement upon, you can say, the scanning tunneling microscope, and hence both of them, they shared half of this Nobel Prize in the field of nanotechnology. Now, when it comes to nanotechnology, there are various different particles, and the first particle, very first nanoparticle to be discovered were fullerenes. All right. So fullerenes were the first particles to be discovered with the help of these microscopes. Now, when it comes to fullerenes, they again showed some remarkable properties and a very unique design, which was again not seen until now. So in 1985, the discovery of fullerenes happened, and 10 years later, 1995, discovery of fullerenes gets a Nobel Prize in chemistry. So another Nobel Prize comes in the way of nanotechnology. So now, in case of fullerenes, this is, you can say, a typical structure of what fullerenes would look like. So fullerenes, as you can see, that it's a football-like structure, correct? You can say that it's a football-like structure where you have these hexagonally joined molecules. So these are all, sorry, atoms. These are all the atoms of carbon. Hmm? So these are all atoms of carbon. So these are all carbon atoms which are joined together, and they make a football-like structure. So this kind of structure is also called as Buckminster fullerene. This is also called as Buckminster fullerene, where we see that there could be 60 odd carbon atoms which are joined together, which have this kind of a structure, football like structure, and hollow from inside altogether, as you can see in this picture. So now discovery of these fullerenes. Why was this discovery important? The discovery was important because of the properties that we saw in fullerenes. We saw that they were very good conductors. We saw that they had very good heat resistance. We saw that they could be used as radical scavengers. We'll discuss about what is radical scavenging at a later stage. But we saw that there were all these properties in fullerenes, properties that we had never seen before. The kind of properties and the efficiency of these properties that we had never seen before. So that's why these fullerenes, they become crucial now. But then after that, in 1993 comes another particle, and this is carbon nanotubes. Now carbon nanotubes, again, if you see, the structures are same, still hexagonal, still joined by carbon atoms. All right, so these things remain the same, but this time, instead of being spherical in shape, they are tube like structures or cylindrical structures, and that's why they are being called as carbon nanotubes. Now in case of carbon nanotubes also, we see that the properties that we saw with carbon nanotubes were exceptional and probably better than the first discovered fullerenes. Better than those fullerenes, we see the properties in carbon nanotubes. And then we realize that there are n number of application areas where carbon nanotubes can be useful. Did carbon nanotubes also get a Nobel Prize? The answer is of course yes. The synthesis and the work around carbon nanotubes also got its Nobel Prize. Now, when you look at the various properties or advantages, it has high thermal and electrical conductivity, optical properties, flexibility, increased stiffness, high tensile strength, they are lightweight, they have the ability to be manipulated yet remaining strong. So that's why we see that there are all these advantages that we see with the properties of carbon nanotubes, and that's why they become useful in a lot of different applications. For example, they become useful in electronics sector. So in electronics sector, they can be used to create very high conductivity. That's why highly efficient wires can be created, or devices that can be created with the help of carbon nanotubes. And what happens is that overall, when we look at these uh, electronic devices, we see that they have very high electrical conductivity, that is the carbon nanotubes, and that's why they can be used to create high speed computer chips, flexible displays, and other electronic devices. So you know that there are foldable display phones that are available now in the market, correct? Many companies have launched it. Samsung has one of the highest selling ones when it comes to it. So this is also where nanotechnology is useful. They use carbon nanotubes, or they use graphene, another substance about which we are going to study. So these kind of substances, they are used in making these foldable displays, the flexible displays. So that's why in electronics, carbon nanotubes are useful. Then in the energy sector, also, they can improve the performance or the efficiency of the energy sources. For example, in creating batteries, they can be useful, because what happens is that they have a high surface area, high electrical conductivity, and that's why they can be used to increase the efficiency of electric energy storage and conversion devices. So that's why we see that in fuel cells, in lithium ion batteries, etc., in solar cells also, carbon nanotubes can be used. Then in biomedical applications, they can also be used, because what we see is that they can be functionalized easily, and they are biocompatible, meaning that they do not have any harmful effects on us in general. So that's why we are saying that they are compatible molecules. So that's why we see that, and also they have unique optical properties that can also be used in diagnostics and imaging applications. So that's why in making various biomedical devices also, we see that carbon nanotubes become useful. Then to make composite materials, because they are very strong, their tensile strength can be almost 100 times better than steel. So they are very strong, yet they are very flexible, not easy to break. So there are all these properties, lightweight at the same time. So that's why in making composite materials, carbon nanotubes can be mixed, or nanotubes can be used to reinforce these materials in the composite material to improve their mechanical and thermal properties. So that's why they can be added to polymers, metals, ceramics, etc., to create strong, lightweight, and heat resistant materials. So that's why in making composite material, because of their strength. Then in environmental applications, we see that they can be used in filtration. They can be used to create water filtration membranes. So they can be used again because they have high surface area and they can efficiently remove the contaminants from water. And then lastly, they also have applications in the aerospace. For example, space technology is where also it can be used. So let's say for when we are making a space vehicle or a launch vehicle, for example. So launch vehicles, there will be a lot of metal parts, but we can replace these metal parts or reinforce these metal parts by making some composite material which is lightweight and strong and thermal resistant all at the same time. So that's why carbon nanotubes can be useful in that sense also. So that's why there are various different ways in which in the future also perhaps we might make the use of carbon nanotubes. For example, in clothing, in making stab proof and bulletproof clothing, they can actually be useful. In making space elevators, a very unique concept on which NASA has been working for, let's say, last almost 30-35 years, where what they want to do is that they want that between the earth station and their international space station, they want to create an elevator using wires which are made of carbon nanotubes. They want to make a lift, they want to make an elevator where things can be moved between the earth and the international space station. So it's a very, very difficult concept, obviously. I mean, it's unthinkable that they'll succeed in it, but they have been working on it extensively. And that's a space elevator as a concept. Every 2-3 years in the newspapers also, it appears somewhere. I remember the first time that I read about it would have been when I was in class sixth, and there was an article. There used to be a newspaper. I mean, there is a newspaper called the Telegraph, which is very famous in Bengal, but also delivered in some of the neighboring regions or neighboring states as well. So, in the Telegraph newspaper, there used to be a magazine every Monday called as Know How. I think Know How now has just become a page or two in the Monday edition, but it used to be an 8-page magazine back then. So everything related to science would be there in that 8-page magazine. A wonderful read. So there was an article based on space elevators back then, and there was a science exhibition in our school. So we presented the model of space elevators based upon whatever we had read. There was very limited or no internet access back then, so we whatever we read, whatever we could understand, we made a model out of it. We showed it in the exhibition, and what we showed, I mean, we also had just by hearted everything. We just went there and showed it. Even the judges who came, they were surprised. They did not have any idea what we were presenting in front of them. So you can say benefit of doubt came to us, and we got the first prize. We won the first prize for that space elevator that we made back then, because neither did we understand what we were presenting, nor did the judges understand what we were presenting. So everyone could not understand. Everyone felt that this is something very complex and unique. Let them have the prize. So that's why there are a lot of ways in which there are uses of carbon nanotubes in the future, where we might see the use of carbon nanotubes, where extensive research and studies are happening as of today. So in making solar panels, in energy storage, in optics, in making radars, in making biofuels, in all these cases, we see that carbon nanotubes can be very, very useful. So but then let me come to a very unique question also that was asked in your 2020 paper. Just read this question. Can you tell me in the comments what you think the answer should be? I'll not launch the poll for this question because there is a reason why I'm not doing that. What according to you is the answer? So I'm getting A, B, C, D. Thankfully, there is no E and F. Otherwise, I would get those answers as well. All right, but a lot of C's and D's, as I see. Hmm. So let me discuss this with you. It's one of the most confusing and complex questions, perhaps, that UPSC has asked. When with sorry, it's a mistake. With reference to carbon nanotubes, consider the following statements. First is they can be used as carriers of drugs and antigens in the human body. Something on the lines of what we discussed of targeted drug deliveries. So targeted drug delivery can be done with the help of carbon nanotubes, because as we discussed that the nanomaterials, they are almost 1,000 times smaller than your blood cells. So that's why they can be carried very easily through the blood, and that's why they can be used as carriers of drugs and antigens. So this is fine. First is correct. The second statement is they can be made into artificial blood capillaries for an injured part of human body. Now, the statement says can be. They can be made. Now, can be, I told you earlier also that can be has immense possibilities. Saying that a can be statement is wrong, you need to have very hard proof to strike this down and say that this is a wrong statement. So, can be anything. And can we make artificial blood capillaries out of carbon nanotubes? Perhaps yes. Have we made it yet? No, we haven't. But there is a branch called as tissue engineering. Tissue engineering. Now, tissue engineering is all about this, that where you create artificial organs or artificial cells and tissues, etc. So that is where making artificial blood capillaries should also be possible. And also remember, we discussed that carbon nanotubes are biocompatible, right? Carbon nanotubes are biocompatible, so we can't even say that these are toxic for our body and hence they can't be used. So they can be. So for me, statement number 2 is correct. Statement 3, they can be used in biochemical sensors. Yes, they can be, and they are already being used in making biochemical sensors. There are multiple of these sensors, especially NASA has been using a lot of these sensors. So carbon nanotubes can be used in doing this. So statement number 3 is also correct. Fourth statement says carbon nanotubes are biodegradable. Now this is where I have some problem. Directly saying that carbon nanotubes are biodegradable is not completely correct, because carbon nanotubes are very stable structures. They do not melt easily, they do not get destroyed easily, they do not decompose easily. It takes a lot of effort to destroy them. So, for example, one of the ways that we use for destroying carbon nanotubes is incineration. Incineration is when you heat something to a very high temperature. So, if you have a material made of carbon nanotubes, you would have to heat it to a temperature of more than 800 degrees Celsius, and then it is something that becomes decomposable, or rather, after that, it gets destroyed. So that's why it's not easy to degrade them. Now what we are doing today is that we are trying to use biotechnology to create certain microorganisms or enhance the properties of microorganisms in a manner that they can act upon carbon nanotubes, so that we can degrade carbon nanotubes, we can destroy carbon nanotubes. But we are making effort. Naturally that's not happening. So saying that they are biodegradable would be natural. It's equivalent of saying that plastic is biodegradable, that you can just degrade plastic, you can just use decomposers or microorganisms and degrade plastic or decompose plastic with it. There are decomposable plastic that now have started to come in the market, but they are made in a very specific way so that microorganisms or decomposers can act on it. But that's not the case with carbon nanotubes. So that's why I wouldn't say that this is a correct statement. For me, this is a wrong statement. Now look at the options and understand why I did not launch the poll. The correct answer should be 1, 2, and 3. The correct answer should be 1, 2, and 3. But UPSC has given this answer. This is the official UPSC answer. See, the best case scenario, I would have said 1, 2, 3, and 4. That even if, let's say, we say that, okay, fine, we know that we can make the microorganisms to degrade carbon nanotubes naturally, I mean, artificially. So let's say that they are biodegradable. But even then, it should be 1, 2, 3, and 4. Saying that 2 is wrong doesn't make sense. Saying that 4 is correct still makes sense. They can still say that, okay, fine, let's consider them to be biodegradable. But there are too many papers going around which are talking about how to make them biodegradable. So saying that naturally they are biodegradable, I wouldn't agree with it. But even if, let's say, I agree with it, statement number 2 is a big problem, where they are saying that they cannot be made into artificial blood capillaries. They cannot be means that not today, not in 50 years, not in 100 years. You can't say that. So maybe they meant something else. Maybe this is a poorly crafted statement. All the possibilities are there. But very the problem more than the question is that it's a very unfortunate question to ask. Why would you ask this question? There's no current affairs here. The concepts are too technical to be asked. These are not basics of nanotechnology. I mean, if it was not asked in the paper, I would never consider anything. I mean, never consider teaching anything related to these statements. But they've asked it. It just looks like that there was somebody who just searched nanotechnology or recent developments in nanotechnology or new papers in nanotechnology. They Googled it and whatever they found, they just gave it in the paper. It looks like that. That it's not a well crafted question, and also it's a very ambiguous or a vague question to ask, first of all, and a very difficult question. How do you expect anybody from outside the scope of nanotechnology to know all this? I mean, somebody coming from biotechnology background, coming from other science background, forget about science background, people from commerce and arts background, how do you expect that those candidates would know anything about any one of these four statements? Only first statement is easy statement. Otherwise, the statements are difficult. Who knows what are biochemical sensors? And even if you know what biochemical sensors are, do you know, or how would you know that whether carbon nanotubes are being used in them or not? So I can go on and on about this question, but such is life that they did ask this question. They should not have, because this is not a technical paper. This is supposed to be a generalist paper. So ask general questions so that people who have diligently prepared for this examination, they should be able to answer the questions. People who have given good amount of time to their preparation, who have completed the static areas, who have completed the current affairs, they've completed the newspapers, they should be able to answer the questions. Even they wouldn't be able to answer these questions, so what's the point? Anyway, let's move on. Otherwise, I have a lot of things to say. I don't want to go to jail. So uh, next is nanowires. Now, in case of nanowires, we see that these are wires which are very small diameters, sometimes as small as 1 nanometer, right? So they are very, very small, and they can be used again in electronic devices. They can be used to make tiny transistors, which can be used for computer chips and in other electronic devices. And at the same time, they become very useful in storage also, because they have very good optical and magnetic properties. So that's why they can become useful in storage, in storing computer data, and all that, doing it in a very, very small space. What we call as miniaturization of technology. Miniaturization of technology. So as the word suggests, that this is where we try to compress or decrease the size of technology. And you have seen this happen, right? Everywhere. The storage, I mean, let's say the computers themselves, they used to be big computers first. I mean, the first computer was a room, and then it kept on shrinking, and now you know that you have laptops, and forget about laptops, you have palm tops, and your mobile phone essentially is a computer. So that's why we have seen miniaturization of technology. Similarly, in storage also, you will see the same, that first it started with bigger devices, the floppy disks, then the CDs, DVDs, then coming to the storage devices like the pen drives, and then now the micro SD cards, which are so small and they have such huge capacities. So that is all called as miniaturization of technology, and nanowires will be very, very useful in doing that, in increasing, uh, you can say, the efficiency of area being used in electronic devices. Then after this we have graphene. Now graphene also is very unique, and more, I mean, more than what is unique about graphene is even the discovery of graphene. If you look at first the structure of graphene, you see that you still have all these hexagonal structures, correct? You still have these hexagonal structures. Still all these are carbon atoms. These are all carbon atoms, but this time not as a cylinder, right? Not as a cylinder, not as a sphere, but rather as a sheet. It's like a two-dimensional sheet of hexagonally arranged carbon atoms, right? What we can say is that it is a two-dimensional sheet of hexagonally arranged carbon atoms. So this is what we can call as graphene. And the same graphene, when stacked one over the other, when you have stacks of graphene one over the other, this is what makes graphite. So this is what makes graphite. So about graphite, we discussed that your pencil, for example, has graphite. And graphite is very easy to break. I mean, pencils, you know that the lead of the pencil that you have inside, it's very easy to break. Why? Because these surfaces of graphene, they can slip over one another. All right, these graphene sheets, they can slip over one another. That's why it's very easy to break. Graphene doesn't break, graphite breaks. All right, graphite breaks, graphene doesn't. Graphene is one of the strongest material that we know of. It is the strongest natural material that has been discovered so far, and it was discovered back in 2004. And in 2004, this discovery also was very unique. All right, this discovery also was very unique. Just give me one moment, please. Sorry. Yeah. So what I was saying is that when you look at graphene, and the discovery of graphene itself has been very unique. The discovery of graphene happened in 2004. All right. Discovery of graphene happened in 2004, and it was a very unique thing that happened. There are two scientists who got the Nobel Prize for discovery of graphene. They got the Nobel Prize in 2010. In 2004, they did the discovery. and they were just playing around. So what happened is, they had graphite, they have the graphite material, and they were using sticky tapes, what you call as cello tapes on it. So they were using the sticky tapes on graphite, they were peeling off the graphite surface, and that is when graphite surfaces, they basically slipped over one another, and the discovery of graphene so happened. So it was, this is what happened, that they just played around with the tape and they won the Nobel Prize. So interestingly, they got the Nobel Prize. There is one Nobel Prize, I don't know whether you know of. It is called as IG Nobel Prize. Do you know of it? Have you heard of it? What is IG Nobel Prize? No. So IG Nobel Prize basically stands for Ignorable Nobel Prize. So Ignorable Nobel Prize is something that is given every year to some of the most useless scientific discoveries and inventions that somebody might have done. All right. And every year this is something that it really happens, that it is basically where this is given, so that uh, I mean, to just honor all the useless discoveries and inventions done in any given year. So ignorable, that's why IG, Ignorable Nobel Prize. Now, one of the discoverers, his name is Andre Gem. Now, this Andre Gem is somebody who has both the Nobel Prizes in his name. He got a Nobel Prize in 1999, sorry, an ignorable Nobel Prize in 1999, and then a real Nobel Prize in 2010. And he's the only one to have done that. There's nobody else who has two Nobel Prizes, one from both the sides. That's somebody who has Nobel Prize, I mean, I, Nobel Prize, that is ignorable Nobel Prize and the actual Nobel Prize. So ignorable Nobel Prize is wonderful. I mean, if you go and see the list of ignorable Nobel Prize of 2023, there are some wonderful Nobel Prizes that you'll see. So, for example, the Nobel Prize in Literature 2023 has been given to the scientists who have studied the sensations that people feel when they use the same word, they repeat the same word many times. So that's the ignorable Nobel Prize in Literature. Very interesting. Oh, the most interesting one is the ignorable Nobel Prize 2023 in Education. It has been given to the scientists who have very carefully studied the boredom that students and teachers feel during a class. So, so the boredom that you might feel, I don't feel. If you feel any boredom during the class, that has been studied and that has gotten the Nobel Prize in Education, Ignorable Nobel Prize in Education 2023. So, um, so Andre Jim is somebody who has both the Nobel Prizes, the actual Nobel Prize for discovery of graphene and the ignorable Nobel Prize for uh, showing magnetic levitation of frog, that how you can use magnetism to make the frog jump. So for that he got the ignorable Nobel Prize. Anyway, coming back to our discussion. So you have graphene, which has arrangement of a single layer of two-dimensional hexagonal lattice of carbon atoms. Now, graphene, interestingly, has been named as a miracle material, or sometimes also called as wonder material. Sometimes also called as wonder material or miracle material. Now, why exactly has it been named so? If you look at all these points, one of the best conductors of electricity, one of the best conductors of heat at room temperature, strongest natural compound discovered, thinnest compound known with just one atom thickness, because it's what? It is a just two-dimensional sheet of carbon. So all these properties together have never been observed, have been never seen in any material thus far. And that's why graphene became so important. Today we find that there are many applications where graphene is being used to enhance the strength of other material, to make LED lighting more efficient and long lasting. It acts as a heat sink in electronic devices. Even we discussed about the foldable devices. So they're foldable displays in electronics. So they can be used in making foldable displays. They're used in batteries and supercapacitors also to make use store more energy and charge faster. And they can also act as barrier between oxygen and water diffusion. So there are a lot of ways. We have found the ways in which graphene can become useful in medical applications, in electronic applications, in everywhere. So that's why graphene became very, very crucial, and that's why we see that there are immense amount of research and development that are happening in graphene, in the field of graphene. For example, this was in the news last year in August. Graphene Aurora program launched at the Maker Village. So now what we see is that a not-for-profit Section 8 company called as India Graphene Engineering and Innovation Center is being set up. And this is something that will try to fill the gap between the research and development and commercialization in graphene. So in case of graphene, that's why what we see is that it will altogether, what we are trying is that there are so much of research that are happening. We need to bridge the gap between the industrial requirements and the research that is happening in this field. So that research is happening according to the requirements of the time, according to what we actually need, right? So that's why this is something that this particular company set up by the government will be trying to do. And that's why it will try to guide, develop, implement, and support the SMEs and startups to commercialize developed graphene technology for scale adoption, so that it can be done at a commercial scale, at a larger scale. So that's why this graphene Aurora program now has been launched by the government of India, because there is so much of research and development happening. For example, CSIR is also actively involved. So in all these cases, we should be able to enhance our knowledge and actually use it for a real time or a real life application. Read this question. C and D are not visible. C is 1, 2, and 4, and D is 1, 2, 3, and 4. Right? This is C, 1, 2, and 4, and this is D, 1, 2, 3, and 4. I'm launching the poll quickly. Please mark your answers. Wonderful. Most of you, 82% of you have marked C, which is the correct answer. So it question says graphene is in use frequently. What is the importance? First, it is a two-dimensional material, has good electrical conductivity. Good is a very basic word. It's one of the best conductors of electricity. It is one of the thinnest but strongest material tested so far. Correct. It is entirely made of silicon. Is it made of silicon? It's made of carbon. So that's why this is wrong. It can be used as conducting electrodes required for touchscreen LCDs and organic LEDs. It is correct that in OLEDs also, you might have heard of OLED TVs, that there are televisions that are made of OLED. There also we make the use of graphene. So correct answer, that's why will be C. Correct answer is C. That is 1, 2, and 4. So this is about graphene. Next is quantum dots, but let's stop here because I also want to take any questions if you have from our discussion on nanotechnology. technology.  。   . 。  . 。  . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . Leaderboard. Let me show you the leaderboard. Yeah, here's the leaderboard. So, congratulations to all of you who have all the correct answers. And for the rest, I hope that you understood where you went wrong. Hmm. All right. So, any questions if you have, please tell me. Are AMOLED displays also made of this? Yes, in AMOLED displays, AMOLED basically stands for active matrix OLED. All right. So, in AMOLED also, at active matrix organic LEDs. So in this also graphene can be used. So yes, graphene can be used in making AMOLED displays as well. Mails, I'll just check when I go back. From May onwards, we have to prepare current affairs for 2025. Uh, mm, Largely, I would say June onwards because May is when the question papers for the last year is also made. So June onwards, but yes, if you have time, ideal is when you take 15 months, that is starting from March, if you can start your current affairs for the next year. Is Byju's going to take science and tech classes this whole year? What is this question? Why would you ask these questions? This is the first time in the new batch that a science class is happening. So if you have problems with these things, don't come to the class. This is very, I mean, be positive, yeah, when you are in the classroom. Ask the questions which are relevant. White background PPTs. I'll check with the team if they can, if it's possible. Why not? We'll do it. Yeah. Any other question? Anyone? So those of you who have, let's say, written to support emails, or if you haven't gotten any reply, please write to me. I mean, please send me those emails or forward me those emails. I'll try to take this up. I'll try to solve the problems. So this is my email ID for that. Any idea which subject next? I think the next subject after me is polity. I'll still have to confirm with the team, but I think it's polity, the next subject. What is the weightage of science biotech? See, weightage could be, you can say, 1 to 2 questions in your prelims examination and 1 question in the mains examination. Books issue we are trying to solve. I've written to the teams also that this is an issue that has been raised by many of you. So we are trying to solve that. I got that from those teams. So let's see. We are trying it. Hmm. When we read from PPTs, white backgrounds are helpful. I'll check with the team. After the class, I'll talk to the team. I'll try to get white background PPTs also uploaded. So any technical issue also, something not opening, etc., please drop an email to the support emails. If they are, if let's say you did not hear from them on time, you can drop an email to me. I'll try to take it up with them. Can I say current affairs is guide for UPSC for framing questions? Yes, in a lot of cases it is. In a lot of cases we do that it is. Hmm. So, all right, so let's pause here. We'll start with the leftover portions of nanotechnology in tomorrow's class. And then after that, we'll start with two smaller topics but important topics of robotics and artificial intelligence in tomorrow's class. All right, so when we come back. So please go back, revise everything. If you have any doubts, please write to us or bring back your doubts tomorrow. For those of you who had written no that you have not attended yesterday's class, please go back and attend those sessions as well. A very crucial portion of biotechnology was covered in yesterday's class. Right? So let's meet back tomorrow. Until then, thank you very much. Good night. Jai Hind.